The broadcast is now starting. All attendees are in listen-only mode.
Good morning and afternoon, everyone. This is Vicki Binger, President of the Coalition Against Childhood Cancer. Welcome to our webinar today, CAC2's May All-Member and Community Webinar. I'd like to do a bit of housekeeping before we get started. First of all, I'd like to thank you for your time and thank our presenters today for making themselves available to us. You may have noticed that the May announcements were full of goodies, and I'd like to just go over a couple of those with you. First of all, we always welcome our new members and thank our renewing members, and today we would also like to very much thank the folks from the broader community who are not CAC2 members who have taken their time to join us today. For all of you who are on the call, I'd like to remind you that we have our summit and annual meeting coming up next month, starting with a dine-around on the evening of Tuesday, June 27th, and continuing with our summit on June 28th and our annual meeting on June 29th, Thursday. It will be held at the Ann and Robert H. Lurie Children's Hospital of Chicago. And I really would like you very much to consider making that part of your summer plans. Both the meeting registration link and the program are available for you on the CAC2 website, and you can see that, that we have this year really a lot of opportunity for member interaction, a lot more time on the schedule for our interest group meetings, and just plenty of opportunities for us to learn from each other. We do have speakers in all of our interest group pillars, but we have a main focus this year on trying to focus on opportunities that we can work together and share and leverage what we're all doing. If you've never attended our June meeting, please consider making this part of your summer plans. Right now, your line is currently muted, but the presenters do welcome questions. If you have a burning question, you may raise your virtual hand at any point during the presentation. We can also receive questions through your dashboard, but I think today it's preferred that you will raise your virtual hand that's on your control panel, and we will call on you. We are recording this session, and we will make it once it's available and post it to our YouTube channel, and have that also. As you see on the screen, we have our CAC2 values. You know that this is the way we start all of our meetings, as a reminder that together we put children and their families first in everything we do, and we do a variety of other things that try to be helpful and supportive of our members and of the childhood cancer community. We try not to compete with our members. We try to be cost-effective, inclusive, collaborative. We try to give voice to the community. We try to stay action-oriented and flexible. One of the reasons that we have this webinar today is because of our member, Gavin Lindbergh, who has been helping guide this webinar forward since the idea came up. Gavin is the president and co-founder of the Evan Foundation, which was established in 2011 in memory of his son, Evan. Evan courageously battled stage 4 neuroblastoma for 4 years before passing at the age of 7 in 2011. This is one of the reasons why we're very interested in the topic today, which is new therapies in neuroblastoma. Evan was the only and special, precious child of Gavin and his wife, Wendy. The Evan Foundation has, in this short amount of time it's been operating, provided over $400,000 worth of support for innovative neuroblastoma research projects, including awards to both of our presenters today. It also sponsors the Evan Lindbergh Neuroblastoma Research Scholars Program at CHOP, which supports talented young investigators interested in neuroblastoma. Besides forwarding a research agenda, Gavin and Wendy spend every Wednesday visiting all the rooms in the inpatient oncology floor at Children's National Medical Center in Washington, D.C., with the Treats and Treasures cart, which brings young patients and families toys, games, books, candies, just fun items to make their hospital stays more pleasant. And the Treat and Treasure cart has made over 6,000 visits since the program started in 2012. Gavin, I guess in terms of thinking about disclosure, is a member of the NANT Parent Advisory Committee, and I think that some of his co-members of that committee are on this webinar today listening. And he was the founding president of the Band of Parents, which has supported neuroblastoma research since 2007. He and his wife Wendy live in Germantown, Maryland. Gavin, I'm going to let you be the host for the rest of the hour.
Well, thank you very much, Vicki, and good afternoon or good morning, as the case may be for folks. I hope everyone can hear me okay. Let me start by adding my thanks to everyone for participating in today's webinar. We're very excited about our program and extremely grateful to our presenters for taking the time to be with us. I think as many of you know, the neuroblastoma research community has been at the forefront of the personalized medicine field, and over 7 years ago, my son Evan participated in one of the first clinical trials designed to treat a specific genetic mutation in his tumor. And the field of personalized or precision medicine for kids with neuroblastoma has grown exponentially since then. So we're very excited today to hear from two distinguished neuroblastoma investigators who are going to highlight their work in this space, Dr. Yael Maseh and Dr. Shahab Asgharzadeh. First, Yael is going to discuss her role exploring, discuss her research exploring the role that the ALK gene plays in neuroblastoma, some of the challenges in effectively targeting ALK, and most importantly, the opportunities that currently exist in utilizing a next-generation ALK inhibitor called daratinib in treating children with relapsed and refractory neuroblastoma. We will hear from Shahab, and I think I actually screwed up the order here. We're going to hear from Shahab first, and he is going to discuss the new approaches to neuroblastoma therapy precision medicine trial that he is leading. And this important study seeks to identify novel genetic immune-related biomarkers and help inform treatment approaches for neuroblastoma families and physicians. Now, after Shahab and Yael are finished, we're going to leave about 15 to 20 minutes for questions and answers. So please, I encourage everyone to take advantage of that time if you have questions or comments for our presenters. Now, before I turn it over to Shahab, I want to say a word or two of introduction about both of our guests. And the trick here, of course, is being concise when the accomplishments on the CVs are so many. So I will do my best, starting with Shahab. He is an associate professor of pediatrics at the University of Southern California, and he is the director of the Neuroblastoma Basic and Translational Research Program at the Children's Hospital of Los Angeles. Shahab received his bachelor's degree in biomedical engineering from Northwestern University and his medical degree from the University of Illinois. He completed his training in pediatric hematology oncology at CHLA prior to joining the faculty of USC in 2005. He is a member of the NIH's Neuroblastoma Target Project, also an active member of the Children's Oncology Group, and he serves on the COG Neuroblastoma Biology Committee. Shahab's research is funded by the NIH, the Department of Defense, and a number of other pediatric cancer organizations. So Shahab, welcome, and thank you again for being with us. Let me also just say a quick word of introduction for Dr. Maseh. Yael is an attending physician at the Children's Hospital of Philadelphia with a special interest in neuroblastoma. She is also an associate professor at the Perelman School of Medicine at the University of Pennsylvania. And I think many of you know the focus of her work and her team's work in the lab at CHOP is the genetics of neuroblastoma. She received her undergraduate degree from Smith College in Massachusetts, a graduate degree from Hebrew University in Jerusalem, and went to medical school at the Sackler School of Medicine in Tel Aviv. Like Shahab, her research is supported by the National Cancer Institute, the Department of Defense, and a variety of pediatric organizations. Yael has numerous awards and recognitions, which I won't go into, except to say that the most important of which is she was my son's favorite doctor. So with that, let me turn it over to Shahab.
Great. Thank you, Gavin, and I appreciate the opportunity to present and appreciate the invitation from Vicki and Gavin to participate in the webinar. I will get to the start this, we'll talk about 20 minutes worth of some of the work we've done within NANT to establish a precision medicine trial. I'm going to go through some general, first I have no disclosures, some general comments, next few slides about what I see as sort of important aspects of precision medicine, what precision medicine is, and how we could sort of teach each other and participate in these trials. So there's a definition that, precision medicine is an emerging approach for disease treatment and prevention taking into account individual variability in genes, environment, and lifestyles for each person. The definition, this is a precision medicine initiative established by NIH as well. Again, mostly focusing on adults, and that's something that we all should sort of try to push our lawmakers to change. Now, there's sort of a paradigm shift over the last 20 years, and that's where this precision medicine initiative came from. And this is, in my mind, divided into sort of three areas. One is methods of diagnosis and prognosis. We've traditionally relied on histology, location of tumor, and size to make both diagnostic and prognostic assessments. And with the advent of genomics, which I'll talk a little bit in detail about, that's now changing. And how we sort of put that into our paradigm, current paradigm, and of diagnosing patients or giving prognostic information becomes very relevant. And Yael's going to talk to the second part of this, which is really changing modalities of our treatments and not discussed in this webinar, supportive care. That's shifting away from the traditional three primary modalities, which is surgery, chemotherapy, and radiation, to these targeted therapies and immunotherapies. There's been a tremendous increase in number of FDA-approved drugs, not for pediatrics, of over about 200 in the last 20 years for various indications. And the last I'll address a little bit as well in this webinar is sort of novel methods to provide not only information about disease burden and reassessment when there's a recurrence, or assessments during the therapy. And this is sort of to complement the current image-based CT MRI that exists in clinical practice today. So germline genomics becomes very important. I don't know a lot of the background of the attendees, but for me, genomics is basically the language of the body, and sort of like a programming language. We could think of all of our organs having a programming language that's in DNA in 4 characters, and we each receive a set of instructions in a book that's written these 4 characters. The books are about 3 billion characters in length, divided into chapters that we call chromosomes, and passages within them that give instructions of how to build certain things, and those passages are called genes. Now, typically we only have 2 copies in each cell in our body, and our different organs utilize this information from these books, one from mother, one from the father, to provide certain functions and abide by certain rules. And there's only 2 copies per cell. And cancer is basically a disease of this genetics and genomics gone completely wrong, where a cell within a particular organ, in my focus in Yael's neuroblastoma, adrenal gland, or cells that are provide an important function in helping our organs function properly, where the genetics is actually perturbed and changed. So they take the book and they do unthinkable things to the book. They take passages and they copy them multiple times, and we call those amplifications. They delete certain passages or regions of the book or chapters, and we call those deletions. They take the passage and they have a paragraph and they change a word. They change one particular character. Remember, it's only 4 characters. When they change a particular character, and that actually changes the meaning of the passage or the function of the passage, we call that a mutation. And advances in the whole genomics field, starting with the Human Genome Project now almost 35 years ago, led to us being able to read the entire book, the genome, by character, and the newest technologies allowed us to take the cancer programming language, or the genome, and actually read those passages as well, and actually figure out which particular word is changed, or what passage is deleted or amplified. And there's lots of verbiage that you hear maybe in the clinical practice as well, or from the physicians, about various techniques. So the whole genome sequencing is where you read the entire book. Every single character that's either in your germline, which is what 2 copies should be within your blood or any other organ that you have that's normal, or within the cancer. So we could actually read the entire book and see whether there's areas that the passages are amplified or copied or deleted, or that there's a verb that's been changed. The exome sequencing really refers to just passages that are important within the 3 billion character document. And those are genes, which are about 20 to 25,000 of them spread out throughout the book. And just look at those particular passages. And gene panel sequencing now says there's certain passages that are really important in cancer, and we'd like to just read those among the 25,000 passages. We may read 100 or 500 of them and look to see whether there's alternative spellings that have changed the meaning of that word. We could also look, and I'm not going to go into detail for these latter 2 parts, RNA species, which is what cells use to get the information transferred from the book into a format that could be used to build proteins, which are the infrastructures of how are used to make a cell function. And these tests are basically, and methylation, last part, methylation is really, if we go with the book analogy, is really just maybe italicizing certain passages so they're not read, or bolding them so they are read. There are different methods that could be used to sort of emphasize a certain passage over others. And these assays that could be used to read this genome or book are done either in pathology labs called CLIA labs, CLIA certified labs, which actually can give information back to the patients, or done in a research setting. And that's, we'll get to what our approach is in the NANS precision trial. And there's also new sort of advances in immune therapy and the role of immune system in tumor microenvironment, saying that the tumor cell is not just a bag of tumor cells, but actually has blood cells, immune cells, and some of these immune cells are actually helping the tumors. So figuring out biomarkers that actually identify which immune cells are actually helping the tumors is really important in also designing novel immunotherapies. And this is gone in adult cancers. Immune checkpoint therapy has become sort of the poster child for these therapies, while in neuroblastoma, we've seen that with combination of therapies, with chemotherapy and dinutuximab, we're achieving some success. Couple of challenges before we get to actually what we do. So we need samples to be able to precisely identify the programming language in the cancer. And there's also ethical and sort of clinical perceptions of rebiopsying children with recurrent disease. And there's a risk-benefit calculus that requires probably a whole seminar by itself. But what I think is important is for people to get educated about when their child is undergoing some sort of biopsy, and again, together with communication with their physicians, think about sort of, what are those biopsies? What is that sample being used? How much sample is being taken? A lot of these techniques require a certain amount of biopsy. There's quality issues to sample processing and preservation techniques that make one particular assay more usable than others. And the issue of when just because you do a biopsy, there may be not enough tumors to actually do some testing, and that's a purity and tumor heterogeneity that becomes involved. And if you need certain amounts of tumor cells within the tumor tissue to be able to conduct some of these assays. So challenges in pediatric cancers, there's really a paucity of recurrent actionable mutations. This is at least in the diagnosis, where we don't see, compared to adult cancers, a lot of mutations in cancer, and we see far fewer in pediatric. This may not be true necessarily in recurrent and relapse tumors, and that's what the NANS Precision trial is actually trying to identify. But we do know that there is an increased prevalence of certain mutations, and ALK being one of them, in recurrent tumors. We really desperately need novel clinical trial designs. We are limited by number of patients, lots of drugs, lots of therapies, how to combine them. We need strong preclinical rationales for doing this work. And we really need all of the data available, and this sort of genomic base of immune biomarkers becomes really important to guide our decision making of how we would design and how we would build these therapies. And last is sort of the novel assays to assess tumor burden and rapidly identify whether these therapies that we're putting children on are successful or not. And there are different ways of doing this. You could look at circulating tumor cells that run in your blood and the bone marrow, or circulating DNA, or even RNA, and I'll touch upon one assay that we've been working on. So this is sort of the diagram of the way I think about, moving forward in neuroblastoma and applicable to many other pediatric cancers. A presentation or a recurrence, we could collect tumor samples and really try to look at some of these molecular techniques, genetics, immune biomarkers, to identify risk groups that are not just purely by a certain number of traditional biomarkers or clinical staging, but by genomic information and tumor microenvironment information, and sort of put them in appropriate combinatorial therapies that have chemotherapy, targeted immune therapies in them. And last, as I mentioned, sensitive biomarkers to assess response. So this is sort of my ideal approach to precision medicine and immunotherapy. Now, a little bit more in details, but basically neuroblastoma can be sort of thought about as a good portion of them having being disease of MYC or MYC family tumors, where we've now identified c-MYC also being very important in the prognosis. On the left shows immunohistochemistry, which is taking tissue slides of tumors and throwing some markers to identify the proteins. And the brown and the red shows that there is either MYC expression or MYC expression, and having those read by a pathologist that says there's high prevalence of those confers poor prognosis in patients. Versus also mutations at diagnosis, ALK being the most common recurrent mutations. There's also ALK amplification, among other ones. But there's a lot of, as you can see, 81% at least don't have a whole lot of recurrent mutations at diagnosis. And we think this is not quite true at relapse. And then we've also, in my group, shown the role of tumor-associated macrophages that when you look at genes that are related to immune system and combine it with some tumor-related genes, allows you to assess prognosis as well. So in the case of a relapse tumor, we have soft tissues that are found only in 50% of the cases at relapse. Also in the bone marrow, where some majority of the tumor would like to sort of go at relapse, there is not a tumor burden. The amount of tumor in the bone marrow is far less than the 30% that people usually think is needed to be able to assess the programming of tumor cells. Because if you take a sample and only 1% of the cells are tumor cells, you could imagine that you don't have enough material to just look at that programming of 1% tumor cells, because it's mixed with the 90% of the genome of the normal cells. And the bone metastasis, again, is another tissue that is problematic in terms of just preserving the tissues in such ways that we could extract the contents or the DNA or the RNA from the bone. And this has to do with sort of the pathology tradition of how we would treat bone, so we could actually look at it under a microscope, but that's not exactly capable of being used for extracting DNA from those tissues. At the bottom is sort of a pie chart of showing from enhanced trial data that combining sort of bone marrow with greater than 1%, majority being less than 30%, and soft tissue, we actually could identify a lot of patients that have some sort of tissue that's accessible. And I think now that the shift has changed to a lot of physicians going after these relapses and actually biopsying them, because we could get information that may be useful for the patients. In our lab, also we've established for the protocol assay where we take those bone marrows in order to enrich them for the tumor cells. So if you can imagine there's 1, 2, or 5% tumor cells, we really want to allow a greater number of patients to participate in these precision studies. So we take a bone marrow sample from a child and we tag it. Let me see if I could, you guys see my arrow? Tag it with antibodies that are magnetically labeled, and you put this through a magnet. The tumor cells that are tagged basically are kept in this column, versus normal cells are taken out, and we could remove the columns or stop the magnet and take out the tumor cells. At the bottom, again, a complicated image, but basically shows on the x-axis the amount of GD2 positive cells. This is a simulation of where we take a tumor cell line and put it in the bone marrow. There's only about 2%, but after enrichment, we're actually getting close to 96% tumor cells, which is more than sufficient, greater than 30% required for doing genetic analyses. So this is the structure of our NANS precision trial. We've established this in almost all of the NANS participating sites. The IRBs are in place. Patients, when they're suspected of having a relapse or recurrent disease, the physicians enrolls the patient on the study and consents the family and the patients to the study to obtain tumor tissue. And the tumor could be either from a soft tissue that's found on a CT scan, a bone biopsy, or a bone marrow biopsy. And these biopsies are obtained and sent over to CHLA pathology, where we look at these tumors, make sure that there's greater than 30% or so tumors in them, and then they're sent to a clinical lab for assessment of 562 genes. It's a gem cancer panel through the Ashion commercial laboratory. And we do immunohistochemistry, which is again looking at the protein markers in the cells for MYC expression, CD163, which is a marker of the macrophages, and PD-L1, which is maybe informative in having patients participate in immune checkpoint trials. We a priori have sort of thought about this, and this is subject to change as the trial progresses, of combining patients into groups of whether they're ALK or have a MAP kinase, which has some targeted treatments for, a metabolic, which is a mostly MYC, MYCN, and some of the cyclin proteins that are involved in cell cycle regulation, and inflammation, which may relate to, again, immune therapy-based trials. A report is generated that includes the gem cancer panel, the immunohistochemistry reports, and our NANS subgrouping and summarizing of all this data. And this is given back to the physician and the patients in hope of having them use this information to make good decisions about, or give them the options to make decisions about what type of trials they want to participate or what type of therapy the child should get. And we've designed a website where we list all the NANS and COG that match these subgroups, as well as those that don't match it. So physicians or the families can go to the website and easily look at all the clinical trials that are available across the country. We also have research aims in the study where we, as I mentioned, we established the assay to enrich the tumor cells. Our goal is to try to look at whether this is feasible, and we could look at DNA, extract DNA from these samples, and perform similar sequencing, next generation sequencing analyses on these samples. And if this comes to fruition and this is successful, then this could be implemented in a clinical assay, which allows more patients to participate in these types of trials. And the last is sort of some of our other work that we're doing with this trial is looking at asking the institutions where the patient had the diagnostic tumor diagnosis performed and getting a diagnostic tumor and comparing the genetic changes that occurs based on therapy from diagnosis to relapse. Look at different sites of relapse. It's been shown in adult literature that there are actually differences in the genomics or the programming of cells that are circulating in your bone marrow or blood versus the ones that are soft tissue tumors. The last is to try to make this also a little bit easier later on is a circulating tumor DNA. We collect serum from the patients, and our hope is that we could actually look at some of the mutations from the plasma from the circulating DNA. I'm going to go through quickly. This is just a snapshot of a report that's given back from Cheyenne, for example, showing there's ALK mutations in this individual with another mutation, and there's FDA-approved drugs as well as potential clinical trials. We're partnering with this group. They'll actually go through clinicaltrials.gov and pick out pediatric trials for this report. This is sort of, again, a listing of the clinical trials that they've put out through their reports. Showing again for ALK, there's several different trials that are ongoing. This is a snapshot of the report that we generate, again, just simplifying it, showing whether the patient belongs to one of those actionable groups or whether there's an actionable gene identified that may have a clinical trial that's not part of this group, and non-actionable genes identified as well, along with whether there's positivity for PD-L1 or the macrophages through the CD163. So far, we've actually enrolled about 15 patients in this trial. It started in November and it's going strong. There's more institutions joining. We have about 10 to 14 day turnaround from the receipt of specimens from the institutions, which I think is very good. There's no tumor board in some of the other trials. People look over this stuff and gather sort of a consortium of people and make some recommendations. Our approach is we want to give this back quickly as possible to the physicians and the patients and provide the tools for them to look at some of the clinical trials that are out there. There's a central database that stores all this genetic information and automated report generation that's been established. This is what you actually see if you go on the NANT.org Precision Study website. Again, those molecular groups that I mentioned are highlighted, which clinical trial, what the drugs may be involved, some comments from us, Kelly Goldsmith, who's a co-chair of the study, and myself, go through some of these clinical trials and annotate them. There's a link to clinicaltrials.gov that people can go and gather more information, as well as some references to whether these are NANT trials or not, seen in almost the lower, the lenalidomide and dinutuximab trial that will be opening at CHLA, at NANT. So this is updated about 3 times a year. We have 16 trials that we've identified that have at least one match to the NANT precision subgroups and 18 trials without any match. And so those are also available for the community to look at. So it's sort of a nice, quick way to look at what trials are available in the COG and NANT institutions. The way we also addressing some of this is we're looking at the germline DNA, and we're reporting the ones that are in the GEM panel of the roughly 600 genes. And if the patients or parents consent to getting this information back, we give this information back with the caveat that they should really go and talk to their physicians and get a clear certified test, because this test is being really done to identify the cancer mutations better, and not necessarily just to provide the germline data. But we provide this is because of American College of Medical Genetics recommendations to report these incidental findings if there's consent. So going back to this, this is paradigm. I'm going to talk quickly about the last part, which is the biomarkers of response. This is the assay, again, that we set up where we take bone marrow from patients, we extract RNA, and have a gene expression profile, again, looking at the genetics that's present in the blood that belongs only to the tumor and cannot belong to anything that's in the normal cell. This has replaced some of the older technology in the left-hand lower corner that we could actually see a tumor cell that's marked by red by proteins that are expressed by that tumor. And then on the right below is the assay detects how quickly you could, the more tumor you have, the more quickly the assay will turn positive. So there's an inverse correlation. The more cells you have on the x-axis is going right, the faster the assay can do its job and give you a report. So that's why there's an inverse correlation. And this is being used in NANT for almost all studies at the disease evaluation through the NANT biology study, as well as all of the therapeutic studies. It's also has significant prognosis. A quick data set is this is patients who are detectable. So by current imaging, if a patient finishes all their therapy, stage 4 patients at the end of the trial, and there's no evidence by CT, MIBG that there's any disease, if there's a positivity in this assay, we could see that it actually confers a worse prognosis for those patients that have a detectable signal in their bone marrow. I just put this last slide for people who are listening to this and want to pursue and look at other precision medicine trials. There's several ones that have been conducted. This is, again, not neuroblastoma specific, but other pediatric cancers and CNS and non-CNS and liquid tumors. And there was a paper that was published, Anita Seibel is the first author recently. And there's obviously also newer trials that are trying to use this and match the mutations found to a particular drug. And the NCI has approached this, other countries, and Yale, I'm hoping, I'm pretty sure we'll also discuss the Fenty trial at CHOP that's using this approach as well. Just a quick thing, a long-term success, almost done. Education of participants, both healthcare providers, physicians need to know about this, the patients and the families, to also inform the healthcare providers who don't know about this. I think these trials are important to return the information to the participants so they can make the best decisions, provide flexibility. We need to establish data sharing with privacy concerns in mind, and transparency about the use of this data for any kind of research. With that, I'm going to thank my lab and the NANT operations who really put this trial together. Lots of collaborators from Atlanta. Kelly Goldsmith is my co-chair. Araz Marshali is the medical director of NANT. Hiro Shimada has been very helpful and instrumental in looking at the tumors that come through, and a slew of other people. And also thank the patients' families who participate in our trials, and foundations who have been supporting this trial, including Evan Foundation, Press On, and the Rising Tide Foundation specifically for this trial. Thank you for your attention.
Thank you very much, Shahab. I think we are going to transition into Yael's presentation, and then we will take time at the end for questions. Yael, please take as much time as you need. Thanks for being with us.
Thanks, Gavin. I realize that we're running a little bit late, and so I'll talk a little bit fast. I wanted to start by thanking you for the opportunity to present some of our work and talk about this exciting trial. Really say that I'm humbled by what you said about Evan, and the reality is that as you well know, we failed Evan and are working very hard to do better for other children and to take the lessons we learned from him and his tumor and apply those in the laboratory to then develop clinical trials that will impact the health of children. So with that, I will briefly talk about how we've come to being on the eve of opening a trial of an exciting next-generation ALK inhibitor.
Hey, Yal.
Yeah.
Yeah, let me just interrupt you real quick and just say, please don't feel like you have to rush. I know everyone who's on the line is very interested in this, so please take your time and, give us all the detail you want. There's absolutely no rush.
Thanks, Gavin. So it was in 2008, after almost 2 decades, that the rare families where neuroblastoma is in fact inherited, just like cancer can be inherited in other contexts, that we identified mutations in this gene ALK, and this was in the germline of families. So the mutation is present in every single cell in the body, and these are the different families that we had been collecting for decades. And you can see in red all the individuals who have that mutation. In black are the individuals who are affected with the disease neuroblastoma. And then we extended this work to characterize really what is the role of this gene ALK in the more common form of neuroblastoma, where there is no family history. And what we found is that in this this portion of the ALK gene, this is kind of a stick figure of what the ALK gene looks like. This part is outside of the cell and this part is inside, and this part is really important. It's called the kinase domain, and when it's turned on through these mutations, it causes abnormal growth of cells and leads to cancers like neuroblastoma. What we showed is that in patients with neuroblastoma whose tumors have an ALK mutation, regardless of whether you have the high-risk form of the disease or the low-risk form of the disease, these kids have an inferior outcome, but certainly the kids who have high-risk neuroblastoma and who have an ALK mutation have an even more inferior outcome. And when we looked within the risk groups, kids with low-risk disease, about 6% have an ALK mutation. 8% of kids with intermediate risk disease have an ALK mutation. 10% have an ALK mutation with high-risk disease, and independently 4% have another way of turning on the ALK gene, which is through what we call amplification, which Shahab mentioned is very similar to how MYC-N is turned on in neuroblastoma. So at diagnosis, using very, very conventional technologies, and we can do a lot better now, but about 14% of kids with high-risk neuroblastoma will have a tumor that relies on the ALK gene for its growth. What we know now is that at relapse, ALK mutations are actually far more prevalent, and we're only starting to really understand why that is and what that frequency is. A recent study that we published shows that the mutation frequency in ALK is as high as 20%. It's probably even closer to 30 or 40%. And that's because we now have better technologies to sequence tumors and have really come to recognize this. I emphasize this because this is why now we talk with families about the role of a biopsy at time of relapse that will provide direct prospect for benefit for children and offer rational treatment options that were not available just a few years ago. So even if the ALK gene is not detected at diagnosis, it is possible that at the time of relapse, we will detect it. So all of the work that we did led to the study that Evan was on, which was run by the Children's Oncology Group. It opened just a year after our initial discovery in August of 2009, and actually it just closed a few months ago. And there we had the opportunity to learn a lot about the very first generation ALK inhibitor. We learned that it's very effective against some tumors that rely on ALK for their growth, like certain subsets of lymphomas. But for neuroblastoma, it became very clear to us early on, and this is one of the major lessons that we learned through Evan, that even though we defined a very high and safe dose of crizotinib that children could tolerate, that was about 200% of the adult dose, that in fact for the many children with neuroblastoma, even with an ALK mutation, this was not going to be sufficient for us to start to really shrink the tumors. And what my lab has done is really gone back to the bench to try to understand why that is. And what we learned is that, first of all, not all of the mutations that we identify in patient samples actually are important. So many of them are, but not all of them are. And then some of them actually are resistant to our ALK inhibitors. And we started to understand why that is. All the mutations that are in blue here, these are the different mutations here, and they sit within this specific pocket in the protein. These ones we think respond to our ALK inhibitors, but these ones in red, they're very different, and they behave differently, and they are more resistant to our ALK inhibitors. And so we've really been very dedicated in the laboratory to develop different treatment strategies to be able to target this gene ALK. And one of the ways that we've done that is we've been still working with crizotinib. It's an important drug. It's been available to us since day one. And we published a paper just about a year and a half ago that when you combine crizotinib with conventional chemotherapy that children with high-risk neuroblastoma get, topotecan cyclophosphamide. And these are 3 different mouse models that have the 3 most common ALK mutations that we see in tumors from patients with this disease. In green is sort of what happens to the mice who get the combination of the chemotherapy and the ALK inhibitor crizotinib. So in this particular model that has a very resistant mutation, you can see the mice treated in red with crizotinib, the tumors keep growing. In purple, these are the mice treated with chemotherapy, and eventually the tumors become resistant. And in green, we see very nice and sustained responses. And this was important work for us because it gave us hope that this drug that we've been developing for so many years could still potentially play an important role. And this really has set the stage for the next phase 3 high-risk neuroblastoma clinical trial, which is on the verge of opening within the Children's Oncology Group, where at diagnosis, all children who are eligible for this study, their tumors will undergo sequencing of the ALK gene, and every child whose tumor has an ALK gene mutation, which we expect will be about 14% of kids, they will be offered the opportunity to get crizotinib in addition to our standard treatment for high-risk neuroblastoma, which, as you well know, is incredibly dose-intensive. We are going to add crizotinib to that therapy. But we're working very hard to identify better ALK inhibitors, and we're really excited about this drug that has this funny number but is now called lorlatinib. And this is really not only a 3rd generation ALK inhibitor, it's really a very potent ALK inhibitor. It's the latest and greatest in terms of the way that it was developed and the way that the structure was made by the biochemists. And remarkably, it was made by Pfizer, who also developed crizotinib. In many ways, they've really led this field because they developed the very, very first ALK inhibitor, crizotinib, and a lot of other companies have developed all these other ones. And then they came in with this newer one, lorlatinib. And what we did in my lab is we compared the activity of crizotinib to this newer drug, lorlatinib, but what we don't show in this paper is that we actually compared it to almost every other ALK inhibitor that's been developed by other companies. And lorlatinib looks, in our lab, really exquisitely potent. So that same model I showed you earlier here that has a resistant ALK mutation, when you treat it with crizotinib, the tumors keep growing. When you just treat it with lorlatinib once a day at a dose that is 10-fold lower than crizotinib, not only do the tumors go away, but they stay away for a long period of time. And we see that even more so in this model where, and this is actually the mutation that Evan's tumor had, where crizotinib seemed effective to us. This is in blue, but you can see by 4 weeks, even though we keep treating the mice, the tumors become resistant, whereas with this drug, lorlatinib, we continue to see a remission, again, at least in the mice. And so we've been really excited about this drug, and so has Pfizer. And in collaboration with them, we've really expedited being able to get this to kids. It has completed phase 1 testing in adults who have ALK-positive lung cancer. It's extremely well tolerated. One of the most exciting features of this drug is that about 1/3 of it goes into the blood-brain barrier. And so there's been dramatic activity seen in lung cancer patients who have, unfortunately, metastases to the brain. And it comes as tablets, but just as Pfizer rose to the occasion many, many years ago, they've made a solution of this for us. And the NANCE study will be the only study that provides access for pediatric patients. So for a number of reasons, the company is not providing this drug through any form of expanded access. It's only available to adults with non-small cell lung cancer. And so we are on the eve of opening the NANT trial. We have very traditional aims to define sort of the highest and safest dose. We're also going to be studied in combination with chemotherapy and really looking early on for responses, because this will be a trial where, I'll go back to this, where you have to have an ALK mutation to be eligible to go on to this trial. And that's because, you know, our data suggests that these are the only patients who are likely to benefit. And so in an era now, as Shahab talked about, of precision medicine, we really shouldn't be wasting the time of families if we don't think that their children are going to benefit from a new drug. One of the exciting things about this NANT trial is that adults will be eligible. So I actually take care of a fair number of adults who have neuroblastoma. And interestingly, we see a much higher frequency of ALK mutations. We don't know why, but in adult neuroblastoma tumors. And so, again, Pfizer has been very supportive of allowing us to provide access to this drug for these rare adults battling a childhood cancer. We're doing a lot of research within the context of this trial to learn more, to do better. So we're doing these quote-unquote liquid biopsies where we'll be collecting blood from kids who are being treated with this drug to see if we can detect the ALK mutation at the time that they start the treatment, and then to follow it throughout their treatment. And we expect that we'll not be able to see it at some point, and if children become resistant to the drug, that we'll be able to detect that early on and even hopefully understand why that is. We are going to be asking for parents' consent to get their tumors a diagnosis and any other tissue so that we can really understand what happens to the ALK gene over time. And patients who have an ALK mutation who've been previously treated with other ALK inhibitors like crizotinib or like ceritinib will be eligible to go on to this trial, although I'm not entirely sure whether these children will benefit as much as the children who've never seen an ALK inhibitor. So to be eligible for this trial, patients have to have an ALK mutation, and that can be done in any clinically approved lab. It can be done through the precision medicine trial that Shahab described, or it can be done through Foundation Medicine. It can be done through, there's so many labs now that offer this kind of testing. Children have to be older than 12, but there's no age cap. As I mentioned, prior treatment with ALK inhibitors is allowed. Patients who have disease in the brain are allowed. Again, we're cautiously optimistic that this will be of benefit. Really, the only major side effect seen in the adult study is this changes in short-term memory. And so we are doing a lot within the NANCE study to assess the neuropsychological function of children who are getting this drug. And so there's going to be questionnaires for parents and questionnaires for kids. Our hope eventually is that this drug will be used frontline, and so we really want to understand whether it has any impact neurologically. And as I mentioned, adults will be eligible to be treated. The first part of the study will be lorlatinib by itself. It's once a day, given by mouth, and we'll be doing the disease assessments after every other cycle, essentially. And it's all outpatient. And then as soon as we define a safe dose of this agent by itself, we're going to be combining it with chemotherapy. We really worked very hard to do everything within the context of one trial rather than first define a safe dose of lorlatinib, then close the trial and write a whole new trial to study it with chemotherapy. We would really like to do everything within the context of one trial. And if it's as exciting in kids as it is in our laboratory, then our hope is to be able to really move this drug forward in patients with newly diagnosed neuroblastoma. So this is just a snapshot of some of the neurocognitive testing that we'll be doing. It differs based on the age of the child, and we'll be having iPads for children to be able to answer some of the questions and do some of the sort of memory testing, hoping that it's pretty user-friendly. We anticipate that this will be open in July, and I do think that this is a very realistic time frame. We've had the usual setbacks, but we're really ready to enroll patients now. And I really want to thank all of the support that we've gotten from this trial, including Pfizer, the Evan Foundation, Cookies for Kids Cancer, Solving Kids Cancer, and their many collaborative partners that I have listed on this slide. And so I will stop there and take any questions you might have.
Well, thank you very much, Yael, and thank you, Shahab, for those presentations. It's really exciting to see the progress that's being made here. And I remember when we were in the fight with Evan, and this opportunity to participate in the crizotinib trial came along really at a time that we were kind of out of hope. And although it didn't work for Evan, the fact that there was hope was important. And it meant that our journey hadn't ended, and we were not only trying to help Evan, but hopefully with Evan's participation in that study, providing some information that eventually is going to help other kids. So thanks to you both for what you do. As we ask around the webinar if there are any questions, let me just start with one real quick to you, Yael, and that is, I guess, two parts regarding the trial you just discussed. Do you have a sense, first of all, as to how many patients you anticipate enrolling? And then secondly, is there an international component to the study as well?
Yeah, thank you very much, Gavin, for those two questions. I forgot to mention that, in fact, our commitment to the development of this drug has allowed us to convince Pfizer and the NANT that this should be an international study. And so the Royal Marsden in the United Kingdom and the Curie Institute in Paris will be special NANT sites that will have this trial open. They may not be set to enroll in July, but they will be set to enroll certainly within this calendar year. And so we're very excited about that, recognizing that there's such a need for this, and these are rare patients, and we really should be working together to develop what looks like an exciting and promising drug. And then we're anticipating enrolling altogether 40 patients, which includes this subset of patients who will get the lorlatinib in combination with chemotherapy. We're actually trying to keep it small. We want to get it done quickly.
Gotcha. And then also, I was wondering if you might say a quick word about some of the ancillary studies that you have planned to try to gather some additional data from the kids who are going to be enrolled.
The most exciting, we're doing a fair bit of correlative studies, and many of them are going to include some of the components that Shahab discussed, but I think for us, the most innovative one that we're doing are these liquid biopsies, and that is in collaboration with Foundation Medicine, where we now have the ability to detect from just 4 or 5 milliliters of blood DNA that's shed by the tumor, so we can actually detect the ALK mutation in the blood of kids whose tumor has an ALK mutation. And that provides us an opportunity to really understand what happens when these kids will get therapy with this new drug. And we're really excited about this. And this will allow us to understand really the depth of response with lorlatinib, and if kids don't respond or develop resistance, why is that, and how can we do better with our next clinical trial?
Terrific. And then,
Gavin, you have a question, if I may? If everybody has just a little more time. Donna Ludwinski, I'm going to open up your microphone and feel free to go ahead and ask your question. Go ahead, Donna.
Okay, thanks, Trish. Thank you, Dr. Mose. This was a fantastic presentation. I had a quick question about your impression about the fact that this is a registration trial. And as far as I know, that's an unusual precedence in pediatric cancer to have a phase 2 single-arm registration trial. So I'm wondering about, is that, you know, a rare occurrence in your experience, and do you know if the pediatric indication is going to be the first indication for this drug that Pfizer is interested in, or is that going to follow the lung cancer indication?
Thanks, Donna. So just to be very clear, so this is not a registration trial, and this is not a phase 2 study either, right? So this is a phase 1 study. We'll have an expansion once we define the highest, safest dose, and but it has been developed in such a way that if the clinical responses that we see in kids are as compelling as we expect them to be, if our science is right, that all of the data could be used for registration and could be submitted to the FDA for an indication in neuroblastoma. But my expectation is that there will be a approval for lung cancer that comes before, you know, any other approval. But this is really the first trial in pediatric oncology where it was designed in such a way that, again, if we see the activity that we hope and expect to see, the data really will be able to be submitted. And having been through this with crizotinib, and I led all of those studies, the data was very, very compelling in kids with anaplastic large cell lymphoma. And, you know, in hindsight, we just never collected the kind of data that the FDA needs for us to get approval for indication for ALCL. So hoping not to make that same mistake again.
I see. That's very interesting. Thank you.
Thank you both. I know that we're running a bit over our time. I have received several questions asking whether this webinar has been recorded. Rest assured it has. It will likely be up late this afternoon, if not by tomorrow morning, on the CAC2 website. So we want to thank you both for sharing that and making it available on the website as well.
And maybe if I could just ask Shahab a question as we start to close here. Shahab, first of all, thank you again for taking the time to be with us. And, you know, you've been a leader in this space for a long time. And, you know, I just wonder, you know, as you look at your current precision medicine trial for neuroblastoma, you know, where you would like to see the field go specific to neuroblastoma. And, you know, as you plan next steps for your study, you know, in a perfect world, you know, how would we take the next step and improve upon, you know, what we're able to do for patients currently?
Sure. Thanks, Evan, for the question. And where I'd like to see this trial go is currently this trial is set up to enroll 60 patients. What I'd like to see is, we give the community information about how we should biopsy children, how much samples should we take to ensure that we just go one time and get the sufficient amount that we need. I really like the ideas, as Yael said, is exploring other options, circulating tumor DNA, gene expression on the blood to assess what are the best techniques to assess patients in phase 1 clinical trials to quickly be able to tell whether it's working or not. And what I'm really pushing also is other immune biomarkers. As you know, my lab is very interested in combination therapies that include immunotherapies. And to me, it's really important for us, as, for example, GD2. The assumption is that every child has a GD2 positive neuroblastoma, but that assumption is actually incorrect. We're developing GD2 biomarkers that we hope to include in that precision and hopefully make it available to all the sites that are treat patients with dinutuximab. Because if you don't have a GD2 positive neuroblastoma, we should not be putting these patients, children with therapies that are painful and require lots of resources and have to have families stay in the hospital for many days. So that's another biomarker that I think is important. That immune biomarkers is where I'd like to push this trial to as well, along with expanding our gene panel testing.
That's terrific. Thank you very much for that. And I think it's safe to say that, you know, folks who have organizations in the neuroblastoma space are very, very interested in being as supportive as we can of this area of research, because it is so promising. So let me just close as we're a few minutes over the hour and say thanks again to Yael and Shahab for taking the time. I want to thank everyone who joined us today. Hopefully you found this as informative as I did. And as Trish mentioned, a recording of the webinar will be up on the CAC2 website by tomorrow, I believe. So with that, thanks again to everyone. And if folks have questions that they want to forward to me or Trish or Vicki, please do so. Thanks again. Take care, everybody.